This video will describe in detail the processes by which substances are moved around a plant. In particular, we'll be describing the steps involved in transpiration and translocation. In addition, we'll aim to explain how a potometer can be used to estimate a plant's rate of transpiration. Water plays a crucial role in the growth and success of any plant. This is due to the fact that water acts as a key reactant in photosynthesis. However, as photosynthesis occurs mainly in the leaves, plants are required to transport any water which they absorb from the soil upwards against the force of gravity. This upward movement presents a challenge. In order to actively move water against gravity, plants would need to expend a huge and unsustainable amount of energy. For this reason, plants have developed a passive approach known as transpiration. Transpiration, which describes the movement of water from the roots to the leaves, uses the natural cohesion which exists between water molecules to create continuous strings of water. To put this more simply, the structure of a water molecule means that it readily forms hydrogen bonds with other water molecules. As a result, we can think of water as sticking to water. This means that as molecules in the leaf evaporate and move out into the surrounding air, they'll pull up the water molecule behind them, which will in turn pull up the next molecule in a continuous sequence. In this way, the plant is able to passively draw water up the stem without having to expend any unnecessary energy. Transpiration then can be thought of as an evolved mechanism to take advantage of a pre-existing problem. Water molecules will always evaporate and move out of the leaf through the stomata. However, by creating these continuous strings of water, the plant can at least use evaporation as a means of driving the transport of water up the shoot. In flowering plants, transpiration occurs through the xylem tissue. The xylem is a hollow, thick-walled tube made up of dead cells. This is a key adaptation as when these cells die, they shrivel up and lose their absorbency. As a result, the walls of the xylem serve only to aid the upward transport of water rather than as an inefficient means of trapping water halfway up the stem. At GCSE, we also note that the cells which make up the xylem have evolved to form one continuous tube. In other words, each individual cell has lost its end walls. Again, this helps to improve the efficiency of transpiration by ensuring that our strings of water molecules have a direct route to the leaves. Now, here's our first pause point question. We've got 3 sentence starters that we need to complete. This will provide a brief description of the process of transpiration. Giving you 2 minutes to copy and complete this description. If I can ask you to pause the video at this point to make sure you have sufficient time to do so. And the answers should be as follows. In other words, transpiration is describing the movement of water from the roots to the leaves. This is crucial because we know that water plays an important role in photosynthesis. Transpiration is a passive process which relies on cohesion between water molecules to pull strings of water up the xylem tissue. We mentioned that one adaptation of the xylem cells was that they are dead. Picking from below the correct option for why this is an adaptive feature. Again, pausing the video, 10 seconds to make your choice. And our correct answer is B. Our xylem cells are dead. This prevents them from absorbing excess water because as they die, they shrivel up and lose their absorbency. So far, we've identified the leaves as the primary site of photosynthesis in green plants. However, any glucose which is produced in these organs must be distributed around all tissues of the plant in order to ensure healthy function. Translocation describes the process by which food molecules are moved from the leaves to the rest of the plant. In order for this to happen, our glucose must first be converted into two types of molecule, sucrose and amino acids. These two types of molecule will be used elsewhere in the plant for energy storage, that is to say they'll accumulate in fruits and tubers. Alternatively, they might be used more immediately in the growth of the plant itself. While transcription occurs through the xylem tissues, translocation takes place in the phloem. As with the xylem, phloem cells are adapted for their particular function. More specifically, these cells are hollow and elongated. This gives them a tube-like structure, allowing large quantities of food molecules to pass through. Unlike these xylem cells, which are missing their end walls entirely, the cells of the phloem exhibit perforated walls at each end. These structures act like a sieve, selectively allowing sucrose and amino acids to pass through, while blocking the movement of larger molecules. Finally, we note that these cells of the phloem are each partnered with a companion cell. These species play an important role in keeping the phloem working as efficiently as possible. Moving then on to our third pause point. Again, we have 3 sentence starters to act as a scaffold for a brief description of translocation, giving 3 minutes to complete this description, pausing your video in order to make sure you've got enough time to complete your work. And answers are as follows. Translocation then is the movement of food molecules from the leaves to the rest of the plant. More specifically, we characterize these food molecules as sucrose and amino acids, which will be used immediately in the growth of the plant, or else will be stored in fruits or tubers. The tissue associated with translocation is our phloem. This tube-like structure is coordinated with companion cells, which help to ensure healthy function of the phloem. At GCSE, we need to feel confident not only with describing the processes of transpiration and translocation, but also in explaining the effect of different environmental factors on their rates. Transpiration, that is the movement of water from the roots to the leaves, is affected by 4 key variables: temperature, wind strength, humidity, and light intensity. More specifically, an increase in temperature causes an increase in the rate of transpiration. This is due to the fact that water in the leaves will evaporate far more readily at high temperatures, pulling more water up the xylem as it does so. Transpiration also increases in windy conditions as a result of water vapor in the air surrounding the leaf being blown away. Humidity, that is the amount of water vapor in the air, also impacts transpiration rate. In more humid conditions, a less steep concentration gradient between the leaf and surrounding air means that water vapor will diffuse out more slowly. As such, transpiration rate decreases with increasing humidity. Finally, we note that increasing light intensity causes an increase in the rate of transpiration. This is due to the fact that in bright conditions the stomata will open widely. While this allows a greater volume of carbon dioxide to move into the leaf for photosynthesis, it also allows a greater volume of water vapor to move out. In each case, we can see that the rate of transpiration is driven by the rate of evaporation through the stomata in the leaves. For this 4th pause point, we can see we have 5 different environmental conditions. For each one, we need to identify whether they will increase or decrease the rate of transpiration in our plant. 1 minute to complete these 5 questions, pausing your video. And answers should be as follows. Now, in actual fact, measuring the transpiration rate of a plant is nearly impossible. While most water loss does occur through the leaves, in order to accurately calculate the amount of transpiration taking place, we'd have to trap and collect all of the water vapour lost through every single tissue of the plant. As this would be far too time-consuming a process, we instead estimate transpiration by measuring the rate at which our plant absorbs water. This process is achieved using a water-filled capillary tube called a potometer. The cut end of a flowering plant is inserted into the capillary via a watertight seal before an air bubble is introduced at the other end. The distance moved by this bubble over a set period of time is recorded, allowing for the calculation of comparable rates between different species or different environmental conditions. The more quickly our air bubble is seen to move along the tube, the more rapid our plant's rate of water uptake, and by proxy, the higher its overall rate of transpiration. Putting this into the context of some exam questions, we first got a model answer. In this question, we're asked to state and explain the change in our rate of movement of our bubble when a lamp is shone onto the plant. In terms of my model answer, I would include the following key pieces of information. So we know that increasing light intensity is going to increase our rate of transpiration. Specifically, we link this with the fact that our stomata will be open more widely when light intensity is higher. What this means is that our plant's rate of uptake of water will also increase in line with our transpiration rate going up. In other words, it means that our bubble will move more rapidly along the capillary tube of our potometer. In this second example, we've been provided with a sentence scaffold to support our completion of the task. The question asks us to state and explain any changes we'd observe if an electrical fan is pointed at the plant. Using your key knowledge from earlier in the video to complete this task in full, pausing your video 3 minutes. Answer should read as follows. We can see then that with increasing wind strength, our rate of transpiration goes up. More specifically, this is because the wind will blow or remove water vapor molecules from the area around the leaf, giving a steeper concentration gradient and causing water vapor to move out more rapidly. If our rate of transpiration goes up, our rate of water uptake will also increase, causing the bubble to move along the length of the potometer more rapidly. In this final example, we are using the prior 2 examples to structure our own independent response. In this case, we are stating and explaining any changes we might anticipate if the plant is placed in a fridge. 3 minutes to complete this answer, pausing your video. Off you go. Our answer should look something like this. In other words, we can see that placing our plant in the fridge decreases the temperature, causing the rate of transpiration to also fall. We can explain this because if our temperature is lower, we'll see a decreased rate of evaporation from the leaves, and if we have less evaporation taking place, then the rate of uptake by the plant's roots will also decrease. This means then, if our rate of uptake has fallen, the bubble will move more slowly through the length of the potometer's capillary tube. In this video, we've described the processes of transpiration and translocation, and in addition, we've used our knowledge of the factors that can affect transpiration to describe any impacts that we might see on a potometer experiment.